Good morning, viewer. Welcome to our FTV English version. Here's a breaking news. Viewers, a centromere is a specific region on a chromosome that ensures that when a cell divides, the chromosome separates accurately so each new cell receives the correct amount of genetic material. Despite their essential role, centromeres remain one of the last major blind spots in the human genome. A recent study published in Nature set out to reveal what had been hidden inside human centromeres using advanced long-read sequencing and custom-built computational tools to piece together 2,110 complete centromeres. There, the researchers sampled individuals from 28 populations groups across 5 continents, then compared their centromeres with 5,747 assembled by the Human Pangenome Reference Consortium to trace how these regions differ and evolve. Viewers, they discovered 226 major centromere haplotypes, distinct genetic patterns, and 1,870 new genetic variants. Yeah, they discovered 226 major centromere haplotypes, distinct genetic patterns, and 1,870 new genetic variants too. Viewers, by studying a four-generation family, they were able to follow centromeres as they changed from parent to child, tracking genetic changes across generation. They discovered that centromeres mutated faster than any other part of the human genome, with certain chromosomes accumulating changes at up to 10 times the rates of others. So, the kinetochore attachment that called the site 2, the anchor point over the chromosome attached to the cell machinery during division, mutated faster than any other part. Viewers, centromeres have often been overlooked in studies of human genetic variation because they consist of long stretches of nearly identical repeated DNA called alpha satellites. Yeah, that similarity makes it difficult for standard methods to distinguish one sequence from another and detect the differences between them. As a result, standard genome references captured a very little of the diversity hidden in centromeres across individual and population, leaving scientists with no clear idea of how centromeres DNA structure and kinetochore placements vary around the world. So they are also in the dark about how quickly these regions mutate or whether their rapid change contributes to chromosomal errors too. For the same, recent advances in DNA sequencing, particularly long read sequencing technology that can finally make sense of complex repetitive regions and structural variants, enable the study to overcome those old technical barriers. Viewers, for the same, in this study, the researchers took advantage of this new technique to explore centromeres and answer some of their questions. Yeah, they began by assembling DNA from 65 diverse individuals using a tool one and the team created called assembly repeat order. They successfully assembled 2110 centromeres and verified that they were more than 99.9999% accurate. To watch how centromere mutations occur in real time across generations, the team turned to a 4 generations, 28-member family, resolving 483 centomeres and comparing parents directly with children. And this allowed them to spot brand new, spontaneous mutations that appeared in just one generation. Amazing, sir. The results revealed that centomeres have far more genetic and structural diversity than previously appreciated. Because when a cell divides, it attaches each chromosome to a protein handle called a kinetochore. For years, scientists assumed there was only one handle per chromosome. So, this sequencing found that some centromeres break that rule. Around 6% have two kinetochores and about 1% have three. Even more importantly, these multi-handed centromeres run in families, passing from parents to children across the generation. BNS exclusive report.